Hello, my name is Estrada, and I am a PhD candidate in the College of Veterinary Medicine, Cellular and Molecular Biosciences program. I will be presenting my research on identifying genes associated with Streptococcus suis disease. Streptococcus suis is a bacterial pathogen that affects the swine industry worldwide. This pathogen causes central nervous system and respiratory disease in pigs and significant economic losses to the swine industry. Some types of strep suis can also cause disease in humans, including sepsis and meningitis. So what is the problem? There is a limited information on characterization of strep suis in swine farms in the United States. This reduces the efficiency of prevention programs for this pathogen. Thus, my research focuses on characterizing strep suis from the United States and identifying genes associated with disease. What are genes, or what are disease-associated genes? Disease-associated genes and their protein products enhance a pathogen's potential to cause disease. They do this in a variety of ways, such as promoting adherence and invasion of host cells, avoidance of the host immune system, and bacterial growth within the host. For my research, I identified these genes using whole genome sequencing. Whole genome sequencing is the process of determining the genome, or the complete detailed genetic information about a microorganism. Comparing the genomes of strep suis from healthy pigs to genomes of strep suis from sick pigs can reveal genetic regions associated with disease. And a similar approach can be used to identify genetic regions associated with disease in humans. The purpose of my research was to identify genes associated with disease in pigs. I compared 208 strep suis genomes recovered from sick and healthy pigs. 8,373 total genes were present among the 208 genomes. Of these, 231 genes were classified as candidate genes associated with disease. That's because they were mostly found in strep suis from sick pigs and absent in genomes from healthy pigs. 17 genes were statistically selected for confirmatory studies. What can we do with this information? The characterization of strep suis and the identification of genes associated with disease aids in the tracking of different types of strep suis in a pig farm over time. It also aids in the identification of strep suis types that are more likely to cause disease in pigs and even in humans. So ultimately, this information will help improve or develop programs for effectively preventing strep suis disease.